Well, as you can see here, it's very important to use words precisely, otherwise you'll lead to tragic misunderstandings. Speaking of tragic misunderstandings, let's go through this slide so we can understand exactly what is being complementary and anti-parallel to what when we're looking at RNA molecules that are being transcribed. So the nomenclature here just gets confusing, so we just have to go through it one step at a time. So what we've got here is a DNA molecule that I've got 5' to 3' on the bottom and 5' to 3' anti-parallel complementary on the top. We're going to transcribe this DNA molecule. Of course, the transcription is going to happen with the RNA molecule 5' to 3', adding new material to the 3' end. Since it is going to be anti-parallel and complementary to its template, that is going to have to be copying as we follow the RNA molecule 5' to 3', but we're going to be walking along the template strand 3' to 5'. And I think that's the confusing bit. That's one of the confusing things about this nomenclature. So this is the template. The blue strand on the top is the template that we're physically going to be copying into RNA. But the RNA molecule, since it is anti-parallel and complementary to that template strand, is actually going to end up having the same sequence as this guy on the bottom, which we're going to then call the coding strand. This doesn't seem right because the coding strand isn't even participating in the reaction. It's just sitting over there waiting to get its template strand re-annealed to it. But since it ends up with the same sequence, we call it the coding strand, and when we look at a DNA molecule in a database, we look at the sequence of the coding strand because it is telling us the sequence of what will eventually be the transcript. So for example, on the coding strand, you'll see that it is 5' to 3' GTACT. The RNA that's being made has that same sequence, except of course we're substituting uracils for thymines, so it has GUACU. So if I want to know what the mRNA is going to look like, I don't look at the template strand, I just look at the coding strand because it is already anti-parallel and complementary to the template strand, just the way the RNA is going to be. So when somebody tells you the coding strand has a particular sequence, like this down here, we know that if we want to know what the sequence of the mRNA is, we just turn the thymines to uracils and we have the same sequence. So the mRNA has the same sequence as the coding strand, DNA. The template strand is, of course, anti-parallel and complementary to that. Now, anytime we take that RNA and we want to make a complement to it, like with a tRNA or with a microRNA that's going to do RNA inhibition, what we need to do then is take the RNA. It's going to be the same kind of base pairing rules, anti-parallel complementarity, and so it's going to go the same anti-parallel complementary sequence, except of course we'll have uracils instead of thymines, and that's going to have the same sequence as the template. So you see down here the template DNA and the complementary RNA are going to have the same sequence after we've substituted uracils for thymines. So really the basic point, the only thing I really want to get across is that when we do Watson-Crick base pairing, we use the same rules and we end up with the same products, whether we're talking about RNA-DNA hybrids, RNA-RNA hybrids, or DNA-DNA hybrids. Watson-Crick base pairing, the straightforward, simple base pairing, is always anti-parallel and complementary. And that was the point. Whenever we do one of these kinds of questions, we get bogged down in this coding strand versus template strand. Hopefully this will help you sort that out.